Can someone confirm you can hear me?
Can hear you.
Mhm. All right. Let me share my screen here. Before we begin, are there any questions? All right, you should be seeing my screen now. Today is March 6th, and we are supposed to be working on chapter 13. Hopefully, we'll finish it. I don't think we will, but we'll be close, hopefully. Let me remind you, your unknown notes pages close March 7th. That's a Friday. The unknown project, the report has to be turned in 11:59 p.m. this Saturday. If you do request a test on Friday, make sure the test, you can get the results within a day. There's only a few tests that allow you to do that. Most of the tests take 2 days or longer. For those of you who haven't finished your unknown notes pages, make sure you do. The unknown notes pages will be graded. It'll be worth 5 points, like a normal lab. And you should have your last test results discussed in your unknown notes pages. Any questions about any of that? My understanding was that the Respondus lockdown browser was not working early yesterday during the quiz. Fortunately, sometime around noon, Respondus lockdown browser started working. And so I only know of one student who wrote in to me and said that they couldn't get in, but hopefully that student did get in. Any questions about anything? All right, let's move on to where we were. I have that we are on this slide talking about viral infection and host immune response. And we did mention that some viral proteins can change, mutate frequently, so that therefore you need to get a vaccine to this virus. Oh, I don't know, every year or every few years. For the flu, the minimum is once every 3 years because the flu virus will change after 3 years so that the vaccine will no longer be very effective. It'll be slightly effective, but not very effective. COVID-19 is another virus that does change fairly rapidly. And the main problem with that is a new strain comes out, which may or may not be different from the previous strain, in which case, if it's different, the last immunization will not work. Other viruses, in fact, most viruses do not change very rapidly. We're still using the polio vaccines made in the 1950s, and they're still good today. So obviously that virus doesn't change very rapidly. Any questions about any of that? All right, so let's talk about viral taxonomy. Viruses are grouped into families. You can group them. What's above family? Let me see now. King Peter's son came over from Germany. So it would be, you can group them into orders. Many viruses are into orders, but much above that, we don't group viruses because virus genomes is not very long and viruses change very rapidly to adapt to their host, meaning their host cells. And so it's very difficult to determine the evolutionary history of viruses. We do group viruses into kingdoms, the kingdom that they infect, but other than that, the higher orders of groupings We just don't group them because we don't know the evolutionary history of viruses. Any question about that? So we do have them grouped into families. Most of them are grouped into an order, but above that, most of the viruses are not grouped except into the kingdom they infect. We can group them into families based on the type of nucleic acid they have, the replication strategy the virus uses, and the viral morphology. All family names end with viridae, like the herpes family of virus, we call them herpes viridae. And then the genus name ends in virus, like human herpes virus. That's the genus. The viruses have different species. This is a group of viruses that share the same genetic information in ecological niche, meaning they're infecting the same host, usually the same host cells. Viruses do not have a scientific Latin name. We simply use common English names to discuss a virus, like the polio virus. That is its scientific name. Any question about any of that? With viruses, we have subspecies, and subspecies are designated by a number. Some species are very important for viruses. A subspecies is sort of like a race when we're talking about humans. The three races of humans. Subspecies would be the same species of virus, but a different subspecies or a different race. And they're very important for a virus. For example, the polio virus subspecies 1 is the polio virus that gives the disease polio. The polio virus subspecies 2 is the virus we use as the live polio vaccine to protect people from getting the disease polio. And that tells you how different the subspecies can be. They're infecting the same people, meaning the same species, probably the same cells as well. So it is the same species, but it's very different in terms of obviously what they cause. One we use for the vaccine, the live polio vaccine, to prevent getting polio, and the other gives us polio. Any questions about viral taxonomy? So I already mentioned the family name ends in viridae. Herpes viridae is an example. The genus would be the herpes simplex virus. So the genus name, the simplex virus. With the herpes viruses, they do have a common scientific name called the human herpes virus. However, this name is not taken hold even in the medical community, and certainly not by the general population. We still call it the human simplex virus, not usually the human herpes virus. The subspecies would be HHV or human herpes virus 1, more commonly called herpes simplex type 1, which causes cold sores. And the other subspecies would be subspecies type 2 or herpes simplex type 2, which tends to cause genital sores. Any question about any of that? That's actually a electron microscopic image of a herpes virus. You can grow viruses in the lab, and when we're growing bacterial viruses, we call them bacteriophages. And bacteriophages are grown inside of bacteria cells. I actually did this. You plate the bacteria so they get a lawn of bacteria growing everywhere. And let me blow this up a little bit. And that's why the plate is mostly, I don't know, white or off-white. It's from the lawn of bacteria growing everywhere. These plaques are when you plate the solution containing a virus, and whenever a virus lands next to a bacteria, the virus can infect the cell, cause the cell to replicate more viruses, and then the virus lyses the bacteria. And then those bacteria, bacteriophages go out to neighboring bacteria cells, infect them, lyse them. And so we get this plaque, which is lyse bacteria. Any question about any of that? So each of these plaques was formed by one or a few viruses that infected a bacteria cell, and then it replicated, infected more cells, and it will grow into something you can see as a plaque. And that would be the color of the agar where that plaque is. Now, some of these plaques are larger than others. This one right here, you can see is actually a three plaques that grew together. But that one is larger, that one is larger, and then this one is small. What probably happened with this large one is that a virus landed on the bacteria cell immediately, and so the virus began replicating immediately. Whereas this one here landed near a bacteria cell. So here's the virus landing here, here's the bacteria cell, and it had to wait for the bacteria cell to grow and then divide until the bacteria cell grew to the virus. And then once the bacteria cell and the virus were touching, the virus could infect the bacteria cell and then start the growth of the plaque. Why there's some smaller plaques and some larger plaques. Any question about any of that? All right. When talking about viral multiplication, you should understand that the viral genome only contains a few of the genes needed for new virion synthesis. For example, the viral gene always contains the capsid proteins that are the capsid coat around the genome of the virus. And how the virus multiplies is it hijacks its host cell and then causes the host cell to replicate the virus. So the virus takes over the host cell's metabolic machinery and then use the metabolic machinery of the cell to make new viruses. This includes using host ribosomes, host tRNA, host amino acids, host ATP, and some of the host enzymes. The basic mechanism of viral multiplication is similar for all viruses, meaning they all hijack a cell, infect the cell. Well, first they infect the cell, hijack the cell, cause the cell to multiply the virus. Bacteria viruses that are called bacteriophages, occasionally they're called just phages. I don't use that term because bacteriophages is a little more clear on what they're infecting. These are viruses that infect bacteria cells. They use two mechanisms for replicating: the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, the host cell is infected, the host cell is lysed, that kills the host cell, releasing the newly replicated viruses. In the lysogenic life cycle, the host cell remains alive for as long as the virus stays in the lysogenic life cycle. Any question about that? All right. To make new bacteriophages, what do we need to make? What does the virus need to synthesize? What components does the virus need? What do you need to replicate if you're going to make new viruses? Well, you have to replicate the viral genome, right? And that happens by, let me blow this up. We're using just double-stranded DNA. You just replicate the viral genome, okay? And then that viral genome will be transcribed to make viral messenger RNA, which will be translated to make viral capsid proteins. And then the viral capsid proteins get together with the replicated viral genome to make the new progeny virions. The lytic life cycle occurs in 5 stages: attachment, penetration, biosynthesis, maturation, and release. Let's watch a little movie on the viral lytic life cycle. I'm not sure this works on my computer.
First, the phage particles.
Let me open that in this one here.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions about that? We'll watch the other half for the lysogenic life cycle in a little bit. All right, the 5 stages of the lytic life cycle are: first, attachment. This is where the phage attaches to the host cell by the tail fibers of the virus. The next stage is penetration. This is where a phage lysozyme, opens the cell wall. The tail sheath then contracts to force the tail contents into the cell. Let me see if I can show you that with the movie here. Oh, here it is.
Acid uses the cell's enzymes and organelles to a certain type of bacteria. Next,
there, that's the virus releasing the enzyme lysozyme, which degrades the cell wall right there.
Phage breaks down the cell wall, and the nucleic acid from this capsule is injected.
The sheath contracts, and that puts pressure on the contents in the head, and that actually injects the viral nucleic acid into the cell. Any questions about any of that part? don't remember where we were. A little further.
multiplication.
Oh, close enough. Once the genome is injected into the cell, it then takes over control of the cell and causes biosynthesis of new virions, where the phage DNA or genome is replicated, and then the phage proteins are made. And then you have maturation. That's the stage where the phage proteins get together with the phage genome to make the newly assembled phage particles. And then the last thing the virus does is it secretes a lysozyme, the enzyme, which is released on the cell wall, which dissolves the cell wall. Water will then move into the bacteria cell, and that will cause the cell membrane to expand until it eventually pops, and that will lyse the cell, causing the new virions that are replicated inside the cell to be released. And that, of course, will kill the host cell. Any questions about any of that? All right, so that's the lytic life cycle, where the virus will attach to the host cell. And how that happens is the adhesions molecules on this part of the tail fiber bind to receptors right here on the cell. And then we have the digestion of the cell wall right here under this portion of the virus. And then the tail contracts, which you can sort of see here. It's reduced compared to there. That puts pressure on the contents of the head, and that injects the nucleic acid into the cell. Two terms you need to know for the lytic life cycle are: one, the burst time. That's the time from infection of the host cell to the time where the host cell bursts. It averages 20 to 40 minutes, depending on the species of virus and the temperature that the cells are grown at. Or you can say the virus is grown at. The burst size is the number of newly replicated virions that are released from the burst cell. This is usually between 50 and 200 virions, depending on the species. Any question about any of that? All right, so that's the lytic life cycle. Let's talk a little bit about the lysogenic life cycle. Upon attachment of the phage and infection of the cell, a lysogenic virus has to decide something right away, whether to stay in the lysogenic life cycle or enter the lytic life cycle. If it enters the lytic life cycle, it just follows the life cycle we just discussed earlier on the lytic life cycle. However, if it enters the lysogenic life cycle, the viral genome then integrates into the host cell's DNA. And then the lysogenic life cycle continues. The phage remains inactive or latent while it is in the lysogenic life cycle. And it remains in the DNA of the host cell, and the two live in symbiosis together as long as the virus remains in the lysogenic life cycle. Any questions about any of that? So here we have the cell becoming infected, lysogenic virus. It injects its genome into the cell. If the cell decides to enter the lytic life cycle, it then just replicates the new virions, bursts the cell, which then will go and infect another cell, starting the life cycle again. However, if the cell decides to enter the lysogenic life cycle, this genome of the virus will integrate into the genome of the host cell. Let me see if I can blow that up a little. And that light purple is the viral genome. The blue purple is the host cell's genome. This is called the prophage, when the viral genome is integrated into the host cell's chromosome. The cell now replicates the viral genome only when the cell replicates its own DNA. Because when you replicate the chromosome here, you obviously replicate the viral genome, the prophage. And that can continue for many cell divisions, where the bacteria and the host stay together in symbiosis in the lysogenic life cycle of the virus. Any question about that part? However, every once in a while, the lysogenic life cycle can end, in which case the prophage becomes excised from the host cell's chromosome, shown here. And then we have the host cell chromosome here with the viral genome there. And then the host cell enters the lytic life cycle, replicating new viruses, the genome and the protein coat, assemble, lyse the virus, infect another bacteria cell, and then once again the life cycle continues. The virus has to decide to enter the lytic life cycle or enter the lysogenic life cycle. Any question about any of that? So let's watch the little video on the lysogenic life cycle.
Lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any question about that? I'll leave it here in case there's a question or want to show something. How do I get out of this? Oh, well, I'll just go over here. So in the lysogenic life cycle, if the virus decides to enter the lysogenic life cycle, the host chromosome integrates into the host cells, not the host genome of the virus, the viral genome integrates in the host cell's chromosome. And we call that a prophage, where the genome of the virus is in the host cell's chromosome. There is a repressor protein of the lysogenic life cycle, and this repressor protein turns off the expression of the genes for the lytic life cycle. So as long as this repressor protein is operating, the cell will remain in the lysogenic life cycle. And then that continues, possibly for many cell divisions, but every once in a while, for reasons that are not fully understood, the lysogenic life cycle ends. Maybe the repressor protein did not shut down the lytic life cycle genes, or something interrupted them. And then the virus will become excised, the genome from the host cell's chromosome. And then once that happens, the virus will enter the lytic life cycle, leaving the lysogenic life cycle. Any questions about any of that? All right. There is one example that we know causes the lysogenic life cycle to leave the lysogenic life cycle and enter the lytic life cycle. And that is if you damage the DNA of the host chromosome, like with a mutagen or something like that, that tends to make the lysogenic life cycle leave the lysogenic life cycle and enter the lytic life cycle. It's like the virus knows the DNA of the host cell has been damaged, and then it doesn't want to risk continuing the lysogenic life cycle. It'll then replicate the virus in the lytic life cycle. But all other reasons for why the lysogenic life cycle leaves the lysogenic life cycle and enters the lytic life cycle, it's not well understood. And they've been studying this now for probably over 60 years, and they still don't have a good handle on why the virus shifts from the lysogenic life cycle to the lytic life cycle. Any question about any of that? All right. There are some consequences of lysogeny. A lysogenic host cell is immune to infection by the same bacteriophage. So if you get a virus, like Lambda virus, infecting a host cell, and it's in the lysogenic life cycle, then other Lambda viruses cannot infect that bacteria cell. And then the second consequence is that phage conversion can occur, and that is where the properties of the host cell can change. And that's because here is the genome of the host cell, and then there is the genome of the host cell with the lysogenic virus in it. It has new DNA, which can express new genes, and those genes can change the phenotype of this cell. And that we call phage conversion, where the host cell exhibits new properties from the genes of the bacteriophage. The best example of this is Corynebacterium diphtherium does not produce the Corynebacteria diphtherium toxin unless the bacteria has a lysogenic virus inside it, meaning the Corynebacterium diphtherium toxin is encoded by the lysogenic virus. Any question about any of that? All right. Enough about bacteriophages. We need to talk a little bit about the multiplication of animal viruses. Viruses that infect animal cells. Remember, people are animals, so that's also human viruses. The multiplication of animal viruses surprisingly follows the basic pattern of bacteriophage multiplication, but there are several differences. There are some differences to attachment. A animal virus attaches to the cell membrane, not to the bacteria cell wall. There's differences in penetration. It's much easier to enter an animal cell than it is a bacteria cell because there's no cell wall you need to get through on the animal cell. Encoding is similar. Biosynthesis is similar. Maturation is also similar, but release of the virus can be different. It can be the same, meaning animal viruses can be released by rupture of the cell, and that occurs by a lytic animal virus. So we do have the lytic life cycle in animal viruses. But the cells can also be released by budding, and this happens by a budding life cycle virus. And these viruses are enveloped. So all budding animal viruses contain an envelope. Usually a lytic virus will be a naked virus. Any question about any of that? All right, the multiplication of animal virus follows the basic pattern of bacteriophage multiplication, but there are several differences. The biggest difference you need to know is an animal virus can replicate either in the cytoplasm or in the nucleus. A bacteria virus or bacteriophage can only replicate in the cytoplasm because that's all there is in a prokaryote cell. But animal viruses may replicate in the nucleus, depending on the species of virus. Any question about any of that? All right, so animal virus multiplication following the basic pattern of phage, bacteriophage multiplication with some differences, such as attachment. There's a difference of attachment. An animal virus only attaches to the cell membrane. If you remember, a bacteriophage attaches to the host cell wall, and then has to degrade that cell wall using the enzyme lysozyme, and then it contracts, injecting the viral genome inside the cell. That doesn't happen with an animal virus. An animal virus attaches to the cell membrane and then gains entry or penetration into the cell either by endocytosis or by fusion. Fusion of the virus envelope to the cell membrane envelope. Does anybody have an earlier version of the textbook? That would be version 1 for the textbook made for Clark College. I'm not sure what version that would be if you have the full textbook, but it's not a late version of the textbook. In the earlier versions of the textbook, it said that penetration of the virus, an animal virus, could happen by pinocytosis. This is incorrect. It happens either by endocytosis or by fusion. Here we're looking at endocytosis, where a virus binds to the cell membrane that causes the cell membrane to begin invaginating further until this is brought into the cell as a vesicle, and then this will pinch off, and so the cell membrane will continue unbroken across here. So that's one way you can bring a virus into the cell. The other way really isn't shown here. This is really showing you endocytosis again, vagination, and then bringing in this vesicle inside the cell, and then that vesicle will be degraded to get at the viral nucleic acid. Let me show you fusion. Here we have our cell with the double membrane. Here we have our virus. and it has an envelope around it. You have to have an envelope if you are having a fusion of the envelope to the cell membrane. The envelope is similar to the membrane of the cell. So what happens is they fuse. And that's actually connected right here. Right there. And then this connects, oops, I gotta break this one too. And then this fuses with that one. And this fuses with that one. Let me bring that out a little better to show that clearer. Now you look at the cell membrane, it goes like this around the virus, and then like that. This opens up so that this viral content can now move into the cytoplasm of the cell. And that's how the virus infects the cell by fusion of the viral envelope to the cell membrane. Any question about any of that? All right, encoding of the virus can happen by viral or host enzymes. Biosynthesis is the production of new viruses. You have to synthesize the nucleic acid of the virus. You have to synthesize the proteins. And then in the next stage, maturation, this is where the nucleic acid of the virus gets together with the proteins to make the newly assembled virions. And then you have release. Release of an animal virus could be the same as a bacteriophage if the animal virus follows the lytic life cycle. And that's rupturing the cell membrane. Usually happens by non-envelope viruses. But if you have an envelope virus, it will be released by the budding. And this virus follows the budding life cycle. So what happens is the newly replicated viral genome comes up and starts pressing on a membrane. This can either be the cell membrane, the nuclear membrane, or the membrane of the endoplasmic reticulum. And the viral genome will come to that membrane where there are viral proteins in the membrane. So where you see these dark blue or in the membrane, those are viral proteins that the virus synthesized and then they get put in the membrane. This could be either the nuclear membrane, the membrane of the endoplasmic reticulum, or the cell membrane. And then the virus begins pushing on that membrane, pushing on the membrane until this rips off, forming a envelope around the virus, and then this will fuse back together, reforming the cell membrane. Any questions about the budding life cycle? Oops, let me shrink that down. Here you're seeing the actual electron microscopic image of a budding virus right here, as well as here, there, and there. The arrow points to a budding virus. A budding virus usually replicates at low numbers so that the virus itself does not usually kill the host cell. Usually, if the host cell dies, it's killed off by the host cell's immune system, recognizing that this host cell is infected. And then the immune system kills it, trying to remove the virus from the body. So a budding life cycle animal virus always has an envelope, and you can see why. That envelope is actually mainly the part of the host cell, a host cell membrane. All right, viruses have different genomes. It can be double-strand DNA, single-strand DNA, double-strand RNA, or single-strand RNA. And if single-strand, you got to say whether it's single-strand positive strand or single-strand negative strand. So each virus types need to have a slightly different mechanism for multiplication and biosynthesis. And we'll talk a little bit about that before we finish our discussion of viruses. Let's turn to talking about the DNA family of viruses. There are 7 family of DNA viruses that cause human disease. So here we have that family name, Poxviridae, Herpes viridae, the strand type, which you don't need to know, the envelope or whether it's naked, which you also don't need to know, the capsid symmetry, which I don't even know, and then the size you don't need to know. You probably should know a little bit about the families if I talk about the family, and then the diseases that the virus family causes, like the Poxviridae family that causes smallpox. Also cowpox, we'll talk a little bit about that. We won't talk about the others. Herpes viridae, we'll talk a little bit about that. We've actually talked a little bit about herpes viridae. This includes herpes simplex virus type 1, herpes simplex virus type 2. I can't read this. The chickenpox virus, which can cause chickenpox or shingles. Epstein-Barr virus, which can cause mononucleosis, Burkitt's lymphoma. I guess that's enough for the herpes viridae. Papilloma viridae, we won't talk about them. Cause warts. Polyoma viridae, won't talk about them. Adenoviridae, we will talk a little bit about adenoviridae. This is a family of viruses that causes the human colds. If you recall, I did mention there are 3 families of viruses that cause the human cold. Adenoviruses, or adenoviridae, is the least common virus to cause the human cold, meaning the third of 3 viruses. So it causes the fewest number of colds. These colds tend to be spread in military barracks. There's no good reason why military life is more likely to spread human colds caused by adenoviridae, but it's thought that the close, cramped quarters, especially a boot camp, and then the stress of boot camp tends to cause people to get human colds, which tend to be caused by adenoviridae. We won't talk about hepaviridae or paraviridae. Like I said, you don't really need to know much of this table, just a few of the families, and then a few of the diseases they cause. You don't need to know all of them. All right, so the DNA containing viruses such as adenovirus and herpes virus, DNA viruses are generally copied in the nucleus using viral enzymes. So a DNA animal virus tends to be copied in the genome. And I don't even know of any exceptions. There probably are a few exceptions, but they're not common viruses, and I bet they don't even infect humans. So when the viral genome is replicated, it'll be translated or transcribed into viral messenger RNA, which will be translated into proteins. The messenger RNA, the viral messenger RNA, will leave the nucleus, go into the cytoplasm to translate the proteins. So as a rule of thumb, if the virus is an animal DNA virus, as a rule of thumb, it's replicating in the nucleus. And I don't know of any exceptions. If it's an RNA containing virus, the rule of thumb is it replicates in the cytoplasm. There is an exception. I'll talk about the exception later. All right, once viral DNA enters the host nucleus, it can either form a provirus where the DNA of the virus integrates into the host cell chromosome, and for a animal virus, this provirus will remain a part of the host DNA for the lifetime of that host cell, meaning that provirus will integrate in the host DNA permanently. It will never be excised. The only time the provirus will be replicated is when the virus replicates the host cell's DNA. Divides into two cells. One cell will get a copy of the provirus, and the other cell will contain the original provirus, or at least one strand of the provirus, if it's a double-stranded DNA. The other thing is that the viral DNA can be expressed, produce new virions, by replicating the DNA, by transcribing the DNA into viral messenger RNA. This happens in the cytoplasm, by the way, this in the nucleoplasm. And then the messenger RNA will be made into the viral capsid proteins. And then the virus needs to mature, where the capsid proteins come together with the viral genome to make the new progeny virions. Any question about any of that? So let's look at the multiplication of a DNA virus in more detail. First, the virus will attach to the host cell membrane. Let me blow this up. Oop, wrong one. What did I do? This is where we are. The virus will attach to the host cell membrane and then will either fuse with the host cell membrane if it's an envelope virus, or it will enter by endocytosis, bringing the viral genome inside the cell. The viral genome will then replicate itself. If it's a double-strand DNA virus, you just separate the double strand into single strands and use one single strand to make a template of the newly replicated viral DNA. The viral DNA will be made into viral messenger RNA, which will then move into the cytoplasm, where protein translation will occur at the ribosome. And then the viruses will mature by the genome of the virus getting together with the protein code of the virus. And then the virus will be released either by budding or by lysing the cell if the cell follows the lytic life cycle. Releasing the virus to begin the process all over again. Any question about DNA viruses? All right, let's talk about RNA family of viruses. There are 15 families of RNA viruses that cause human disease. You only need to know a few of the families if we talk about them, and then the diseases they cause, the rightmost column here. Picornaviridae is a very big and diverse family of viruses that, among other things, cause the disease polio, hepatitis A, and then the disease we call the human common cold. So Picornaviridae is causing a lot of different diseases. They can be very different diseases. Like polio is much more severe than the human cold. The family Picornaviridae is the #1 family of viruses that cause the human cold, meaning most colds are caused by this family. So this is the #1 family causing the human cold. Calciviridae, we won't talk about. Hepiviridae, we won't talk about. Astroviridae, we won't talk about. Togoviridae, we're not going to really talk about it, but you might want to know that this is the virus that causes rubella, as well as encephalitis. Flaviviridae, we really won't talk about. Coronaviridae, I'll talk about it in 3 slides. This is the virus that family that encompasses COVID-19. Retroviridae is the exception of the RNA family of viruses. All the other RNA viruses, family of viruses that cause human disease, all of them except for Retroviridae replicate in the cytoplasm. So as a rule of thumb, if the virus contains RNA as its genome, it replicates in the host cell's cytoplasm. And if it contains DNA, we've already mentioned that it replicates in the nucleus. Retroviridae is an exception, though. This is the one family of RNA viruses that actually replicate in the nucleus. And once I talk about that, it'll be clear why the family Retroviridae replicates in the nucleus. Paramyxoviridae, I'm not going to talk about, but you should probably know that this family causes a whole lot of common childhood illnesses, such as, hepatitis, the mumps virus, measles. Rhabdoviridae, we're not going to talk about it. Flaviviridae or Filiviridae, we're not going to talk about that. Orthomyxoviridae, I will talk just briefly about that. And Orthomyxoviridae is the family of viruses that cause the human flu or influenza. And that's probably all you need to know about that. Buniviridae, we won't talk about. Arenoviridae, we won't talk about. Rioviridae, we won't talk about. Any questions about the RNA family of viruses? Here we're looking at some RNA family of viruses, and you can note there are different shapes, different sizes. Like this is a real small cell because that's blown up 10 times compared to these 2 cells. Oh, wrong one. Replication of RNA family of viruses is essentially the same as for DNA family of viruses. There are some differences because we have a different genome. This genome is RNA instead of being a DNA genome. Viral replication, already mentioned as a rule of thumb, happens in the cytoplasm because that's where the machinery is for replicating RNA. However, remember there is one exception, and that is the family Retroviridae doesn't replicate in the cytoplasm, even though it's an RNA family of viruses. Herpes, no. Retroviridae replicates in the nucleus. And that'll be clear once we talk about family Retroviridae, which probably we won't get to today. We'll see. Some examples of an RNA family of virus. If it's a double-stranded RNA family, you don't need to state anything further. But if it's a single-strand RNA virus, you need to state whether that single strand of RNA genome is the positive strand. That's the strand of RNA that the ribosome can read. So the positive strand will have AUG in it. The ribosome can translate that viral genome into viral proteins. There's also the negative strand genome of RNA animal viruses. The negative strand is antisense to the positive strand. So the ribosome cannot read the negative strand genome. To replicate or translate this viral genome, you have to make the positive strand, and then you can use the positive strand to translate the viral genome into viral protein, as well as you need the positive strand make to replicate more negative strand RNA. Any question about any of that? Hopefully I said that correctly and clearly enough. Oops, get out of there. So if the genome is DNA, it's very simple. You just need to transcribe one of the DNA strands to make the messenger RNA. And then that messenger RNA can be read by the ribosomes and translated into protein. And that is if the, well, we're talking about DNA, so that's all right. If, on the other hand, the genome is positive strand RNA or double strand RNA virus, so positive single strand RNA genome or a double strand RNA genome, how you make more of the positive strand genome is first you make the negative strand, and it'll be complementary to the positive strand. And then you use the negative strand as a template to make more of the positive strand genome. And then that positive strand genome can be read by the ribosomes and translated into viral proteins. Any question about any of that? Now, if you have a negative strand RNA virus and it's single strand, then you need to make the positive single strand RNA virus and then use that strand as a template to make more of the negative strand genome. Any question about any of that? So note that the negative strand RNA can never be translated into protein. So for an RNA negative strand, single strand virus to replicate, we need to first make a positive single strand RNA. Then we use this as the template to make more of the negative single strand RNA. We use the positive single strand RNA in translation to make the capsid proteins, which will then self-assemble with the RNA genome to make the new progeny virions. Any question about this? If, on the other hand, the animal virus is a positive single-strand RNA virus, you first make the negative single-strand RNA virus, and then you read this to make the new genome, which in this case would be either DNA or positive strand RNA. And then you use the positive single-strand RNA to make the capsid proteins in protein translation. Once again, you get a union of the genome and the capsid proteins, and that will make the new progeny virions. Any question about this slide? All right, positive single-strand RNA virus multiplication, a little more detail. First, we have a virus which binds to the host cell membrane, and it will either gain entry into the cell by endocytosis, or if this is an envelope virus, it can gain entry into the cell by fusing the viral envelope to the cell membrane. Once the virus is inside the cell, it'll be the capsid proteins will be degraded, releasing the viral genome. Since we're talking about a positive single-strand RNA virus, that'll be multiplied by first making the negative strand single-strand RNA virus, and then using that negative strand as a template to make more of the positive single-strand RNA. Then the newly replicated virions will self-assemble, where the protein coats of the virus will self-assemble around the nucleic acid of the viruses. In the lytic life cycle, this infected cell will then lyse, releasing the newly replicated progeny virions. However, the leaving of the cell can also happen by budding, where the virus will actually bud out of the membrane of the cell and then begin the process of viral multiplication once again. Any question about positive single-strand RNA virus multiplication? All right, let's talk a little bit about antisense RNA virus multiplication. You have the virus, once again, binding to the cell, gaining entry to the cell, either by fusion of the membrane, of the envelope of the virus to the cell membrane, or by endocytosis. The negative sense anti-strand RNA can replicate itself by making positive strand RNA and then using the positive strand RNA to make more copies of the negative single strand RNA virus. How you get the capsid proteins being made is you get the positive antisense strand RNA to make more virions, more RNA, which can be used to, if it's positive strand, which in this case would be the replication of the negative strand, and then that can be read by the ribosome to translate the positive antisense single-strand RNA into protein translation at the ribosome, making the protein code of the viruses, which will then mature by the protein coat getting together with the viral genome, which will then be released from the cell either by lysis, if it's the lytic life cycle, or by budding, if it's the budding life cycle. Any question about any of that? All right, let's talk about the family Retroviridae, the retroviruses. These are RNA family, a family of RNA containing viruses, but they replicate in the nucleus, not in the cytoplasm. So this is the exception to the rule of thumb, where an RNA virus will replicate in the cytoplasm. The retroviruses include the lentiviruses, which include HIV-1 and HIV-2, both of which cause human AIDS, as well as feline leukemia virus, which causes leukemia in the cat, which then weakens the cat's immune system, and it's called kitty AIDS because the kitty will now become subjective to many different infections, and the patient won't have any, if a little, if any, immunity, and so that's why it's called kitty AIDS, feline leukemia virus. It doesn't cause AIDS, it just weakens the host immune system, so the kitty is much more likely to get an infection by a microbe. Retroviruses are unique in that they contain the gene for the enzyme reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase. It can make DNA from an RNA template. Let me show you that. Retrovirus, we have a single strand, positive strand RNA in the virus genome. And in fact, we have two of those. That's not terribly important, but in the Retroviridae, you always have two of the same strands of RNA. Anyways, let's write that out. That's positive single strand RNA. And then you can have reverse transcriptase. Let me get an arrow here. I'm just going to put RT for reverse transcriptase. And that makes single strand DNA. This. That single strand DNA can be acted upon by reverse transcriptase to make double strand DNA. This isn't discussed in your textbook, but reverse transcriptase actually takes two steps to make double strand DNA. first making single-strand DNA from RNA, and then making double-strand DNA from the single-strand DNA. Oops. This double-strand DNA then integrates into the host cell chromosome and becomes a permanent fixture of the DNA of that host cell. For as long as that host cell is alive, it'll contain retrovirus DNA in the host cell. That's one reason why if a patient becomes free of the HIV virus, you never know if it may come out later because the patient may have a cell in them with, I guess I should make this another one here. You never know if the AIDS patient has a cell in them that contains the HIV DNA or genome. And this could, let's go here, be transcribed, in which case you can make viral messenger RNA, which is positive. I guess I should mention that too. So this is positive. Yep. Positive single strand RNA, which is the viral genome. Anyways, the viral messenger RNA can be translated into viral proteins. Whoops. Together with the viral genome. Let's go ahead and do that one. To make the new virions. Retroviruses are always enveloped, meaning they always follow the budding life cycle, at least when they're replicating. I'll talk about what I'm talking about in just a little. Well, I guess I'll say now that the budding life cycle virus may enter the, gosh, what's the name of that life cycle virus? It may enter the, not the lytic life cycle, but it may enter a life cycle where the virus is not actively replicating, the virus is not causing disease, and I'm losing my mind for what the heck that's called. Anyways, it could do that, but obviously if it's budding out, this can be then releasing an HIV virus or another herpes virus. Any question about any of that? Let me see if we have anything further to discuss. I think I'll stop here and we're gonna continue with the Family Retroviridae next time. Meaning I don't have enough time to finish talking about Family Retroviridae. Any questions about any of that? All right, if there's no questions, I will be in the lab only for 15 minutes to answer any questions. If you don't have any questions, you do not need to show up in the lab. All right, goodbye.